Today's lecture on cytological basis and molecular mechanism of crossing over. First, let's have a look at the objectives. To prepare the students about linkage and crossing over, to teach them about the molecular mechanism of crossing over, to teach them about the interference and coincidence. Dear students, let us first understand linkage and crossing over. Eukaryotic organisms contain a linear chromosome having a very long segment of DNA. Each chromosome contains many individual functional units which are called genes which influence an organism's traits. A typical chromosome contains hundreds or few thousand different genes. When two or more genes are located on the same chromosome, they are said to be syntentic. Genes that are syntentic are physically linked to each other because each eukaryotic chromosome contains a single continuous linear molecule of DNA. Genetic linkage is the phenomenon in which genes that are close together on the same chromosome tend to be transmitted as a unit. Therefore, inheritance patterns get altered by genetic linkages. Since a chromosome contains a group of genes that are physically linked together, therefore, chromosomes are sometimes called linkage groups. In species that have been characterized genetically, the number of linkage groups equals the number of chromosome types. For example, human somatic cells have 46 chromosomes which are composed of 22 types of autosomes that come in pairs plus one pair of sex chromosomes, the X and Y. Therefore, humans have 22 autosomal linkage groups and an X chromosome linkage group, and males have a Y chromosome linkage group. In addition, the human mitochondrial genome is another linkage group. Crossing over. Crossing over is a phenomenon which involves a physical exchange between the chromosomes. The exchange events occur during the prophase of the first meiotic division when duplicated chromosomes have paired. Although 4 homologous chromatids are present, forming what is called a tetrad, only 2 chromatids cross over at any one point. Each of these chromatids breaks at the site of crossover and the resulting pieces reattach to produce the recombinants. The other 2 chromatids are not recombinant at this site. Each crossover event therefore produces 2 recombinant chromatids among a total of 4. Only 2 chromatids are involved in an exchange at any one point. However, the other 2 chromatids may cross over at a different point. Thus, there is a possibility for multiple exchanges in a tetrad of chromatids. There may, for example, be 2, 3 or even 4 separate exchanges typically called double, triple or quadruple crossovers. Note, however, that an exchange between sister chromatids does not produce genetic recombinants because the sister chromatids are identical. Crossing over is the reciprocal physical exchange of chromosome segments at corresponding positions along pairs of homologous chromosomes. The positions at which crossing over occurs along the chromosomes are largely random and vary from one meiosis to another. The physical exchange that occurs in crossing over is facilitated by the alignment of the homologous chromosomes brought about by the synaptonemal complex. If there are genetic differences between the homologues, crossing over can produce new gene combinations in a chromatid. There is usually no loss or addition of genetic material to either chromosome since crossing over involves reciprocal exchanges. A chromosome that emerges from meiosis with a combination of alleles that differs from the combination with which it started is called a recombinant chromosome. Therefore, crossing over is a mechanism that can give rise to genetic recombination. At the end of pachynemia, the synaptonemal complex is disassembled and the chromosomes have started to elongate. Now, let's move on to cytological basis of crossover. The cytological evidence for crossing over can be seen during late prophase of the first meiotic division when the chiasmata become clearly visible. Chiasmata are simply traces of the actual exchange process. Chromatids that have experienced an exchange probably remain entangled with each other during most of the prophase. Eventually, these entanglements are resolved and the chromatids are separated by the meiotic spindle apparatus to opposite poles of the cell. Therefore, each chiasmata represents an entanglement that was created by a crossover event earlier in prophase. At this time, paired chromosomes repel each other slightly, maintaining close contact only at the centromere and at each chiasma. This partial separation makes it possible to count the chiasmata accurately. As we might expect, large chromosomes typically have more chiasmata than small chromosomes. Thus, the number of chiasmata is roughly proportional to the chromosome length. The appearance of chiasmata late in the first meiotic prophase might imply that it is then that crossing over occurs. However, evidence from several different experiments suggests that it occurs earlier. The event responsible for recombination, namely crossing over, occurs rather early in the meiotic prophase. Although almost all the DNA is synthesized during the interphase that precedes the onset of meiosis, a small amount is made during the first meiotic prophase. This limited DNA synthesis has been interpreted as part of a process to repair broken chromatids, which is thought to be associated with crossing over. Careful timing experiments have shown that this DNA synthesis occurs in early to mid prophase but not later. The accumulated evidence therefore suggests that crossing over occurs in early to mid prophase long before the chiasmata can be seen. Most biologists believe that chiasmata are merely vestiges of actual exchange process. Chromatids that have experienced an exchange probably remain entangled with each other during most of the prophase. Eventually, these entanglements are resolved and the chromatids are separated by the meiotic spindle apparatus to opposite poles of the cell. Therefore, each chiasma probably represents an entanglement that was created by a crossover event earlier in prophase. But why do these entanglements occur at all? Many biologists believe that the entanglements created by crossing over are a way of holding the members of a bivalent together during prophase 1. In some organisms, prophase 1 is protracted. In human females, for example, it may last as long as 40 years. Without crossovers, paired homologues might accidentally separate from each other during this long time, and homologues that have separated might not disjoin properly during the ensuing anaphase. Faulty disjunction of the chromosomes during the first meiotic division would ultimately lead to aneuploid gametes. Thus, crossing over appears to be a mechanism to hold paired homologues together so that when division does occur, the homologues are distributed appropriately to each of the daughter cells. In this way, then the possibility for non-disjunction is minimized and aneuploidy in the gametes is largely prevented. Now, moving on to molecular mechanism of crossing over. Crossing over involves an exchange between DNA segments that are similar or identical in their DNA sequences. Eukaryotic chromosomes have similar or identical sequences frequently participate in crossing over during meiosis 1 and occasionally during mitosis. Crossing over involves the alignment of a pair of homologous chromosomes followed by the breakage of two chromatids at analogous locations and the subsequent exchange of the corresponding segments. There are two types of crossing over that may occur between replicated chromosomes in a diploid species. When crossing over takes place between sister chromatids, the process is called sister chromatid exchange (SCE). Because sister chromatids are genetically identical, SCE does not produce a new combination of alleles. By comparison, it is common for crossing over to occur between homologous chromosomes during meiosis. This form of homologous recombination may produce a new combination of alleles in the resulting chromosomes. In this second case, homologous recombination has resulted in genetic recombination, which refers to the shuffling of genetic material to create a new genetic combination that differs from the original. Homologous recombination is an important mechanism to foster genetic recombination. Many of the popular models of crossing over were derived from a model proposed by Robin Holliday in 1964. Holliday's model was one of the first that explained most of the genetic data available at the time by a mechanism involving the breakage, reunion, and repair of DNA molecules. This mechanism, like many others that have been invoked, begins when an endonuclease cleaves single strands of each of the two parental DNA molecules. Segments of the single strands on one side of each cut are then displaced from their complementary strands with the aid of DNA helicases and single strand binding proteins. The helicases unwind the two strands of DNA in the region adjacent to the single strand incisions. E. coli contains both an endonuclease activity that makes single strand breaks in DNA and a DNA helicase activity that unwinds the complementary strands of DNA in the region adjacent to each nick. The displaced single strands then exchange pairing partners, base pairing with the intact complementary strands of the homologous chromosomes. This process is stimulated by proteins like E. coli Rec-A protein. Rec-A type proteins have been characterized in many species, both prokaryotic and eukaryotic. Rec-A protein and its homologues stimulate single strand assimilation, a process by which a single strand of DNA displaces its homologue in a DNA double helix. Rec-A type proteins promote reciprocal exchanges of DNA single strands between two DNA double helices in two steps. In first step, a single strand of one double helix is assimilated by a second homologous double helix, displacing the identical or homologous strand and base pairing with the complementary strand. In the second step, the displaced single strand is similarly assimilated by the first double helix. The Rec-A protein mediates these exchanges by binding to the unpaired strand of DNA, aiding in the search for a homologous DNA sequence, and once a homologous double helix is found, promoting the replacement of one strand with the unpaired strand. If complementary sequences already exist as single strands, the presence of Rec-A protein increases the rate of renaturation by over 50-fold. The cleaved strands are then covalently joined in new combinations by DNA ligase. If the original breaks in the two strands do not occur at exactly the same site in the two homologues, some tailoring will be required before DNA ligase can catalyze the reunion step. This tailoring involves the excision of nucleotides by an exonuclease and repair synthesis by a DNA polymerase. The sequence of events described so far will produce X-shaped recombination intermediates called chi-forms, which have been observed by electron microscopy in several species. The chi-forms are resolved by enzyme-catalyzed breakage and rejoining of the complementary DNA strands to produce two recombinant DNA molecules. Dear students, the next part is recombination frequency as a measure of linkage intensity. Genetic mapping allows us to estimate the relative distances between linked genes based on the likelihood that a crossover will occur between them. If two genes are very close together on the same chromosome, a crossover is unlikely to begin in the region between them. However, if two genes are very far apart, a crossover is more likely to be initiated in this region and thereby recombine the alleles of the two genes. Experimentally, the basis for genetic mapping is that the percentage of recombinant offspring is correlated with the distance between two genes. If two genes are far apart, many recombinant offspring will be produced. However, if two genes are close together, very few recombinant offspring will be observed. To interpret a genetic mapping experiment, the experimenter must know if the characteristics of an offspring are due to crossing over during meiosis in a parent. This is accomplished by conducting a test cross. Most test crosses are between an individual that is heterozygous for two or more genes and an individual that is recessive and homozygous for the same genes. The goal of the test cross is to determine if recombination has occurred during meiosis in the heterozygous parent. Thus, genetic mapping is based on the level of recombination that occurs in just one parent, the heterozygote. In a test cross, new combination of alleles cannot occur in the gametes of the other parent which is homozygous for these genes. Now moving on to 2-factor and 3-factor crosses. Mendel also analyzed a number of crosses in which two pairs of alternative traits were involved simultaneously. In each case, he obtained the same results. From these experiments, he proposed his second law, the principle of independent assortment, which states that the factors for different pairs of traits assort independently of one another. In modern terms, this means that pairs of alleles for genes on different chromosomes segregate independently in the formation of gametes. Consider an example involving the pair of trait for seed shape, smooth and wrinkled, and the pair of traits for seed color, yellow and green. Yellow is dominant to green. When Mendel made crosses between true breeding smooth yellow plants, capital S, capital S, capital Y, and wrinkled green plants, small s, small y, all the seeds from this cross were smooth and yellow, and the results of the monohybrid crosses predicted. The smooth yellow parent produces only gametes which give rise to zygotes upon fusion with the gametes from the wrinkled green parent. Because of the dominance of the smooth and yellow alleles, all F1 seeds are smooth and yellow. The F1 are heterozygous for two pairs of alleles at two different loci. Such individuals are called dihybrids, and a cross between two of these dihybrids of the same type is called a dihybrid cross or 2-factor cross. In other words, dihybrid crosses are the crosses in which we follow the inheritance of two different characters within the same groups of individuals. For example, an experiment in which one of the characters is seed shape found in round or wrinkled variants, the second character is seed color which exists as yellow and green variants. Mendel also confirmed his laws for three pairs of traits segregating in other crosses. Such crosses are called trihybrid crosses. Here, the proportions of F2 genotypes and phenotypes are predicted with precisely the same logic used before, by considering each trait independently. The independently assorting pairs of traits in the cross are smooth and wrinkled seeds, yellow and green seeds, and purple and white flowers. There are 64 combinations of 8 maternal and 8 paternal gametes. Combination of these gametes gives rise to 27 different genotypes and 8 different phenotypes in the F2 generation. The phenotypic ratio in the F2 is 27:9:9:9:3:3:3:3:1. So, a 3-factor cross is a cross in which 3 characters of the organism are under consideration. For example, an experiment in which one of the characteristics is seed shape, found in round or wrinkled variants, the second character is seed color, which exists as yellow and green variants, and the third character is the color of the flowers, which may be purple and white. Moving on to the next part, that is interference and coincidence. Interference is a phenomenon in which the occurrence of a crossover in one region of the chromosome decreases the probability that a second crossover will occur nearby. In other words, the first crossover interferes with the ability to form a second crossover in the immediate vicinity. To provide interference with a quantitative value, we first calculate the coefficient of coincidence C, which gives us the extent of interference and is calculated as the ratio of the observed number of double crossovers to the expected number. The equation is C coincidence is equal to observed number of double crossovers divided by the expected number of double crossovers. Then the interference I is expressed as I is equal to 1 minus C. Now moving on to somatic cell genetics, an alternative approach to gene mapping. Mammalian gene mapping techniques are now sufficiently advanced to contribute significantly to human molecular genetics. Restriction fragment mapping can be used to place polymorphic genetic markers at random sites within the genome, and these sites used to assign genes responsible for disease conditions to a chromosomal region. Somatic cell genetic techniques can then be applied to saturate that region with additional restriction fragment markers, some of which will be closely linked to the disease gene. Closely linked restriction fragment markers, especially flanking pairs of markers, can act as predictors for the transmission of defective genes to offspring. A series of tightly linked flanking restriction markers might in addition contribute to the eventual isolation and cloning of the diseased gene itself. That was all about today's topic. Hope you have understood. Thank you.